Reminder, there's a biology colloquium today at noon up in CST 300. It's a biology colloquium, but it will be tailored for the general public, but it will not be a discussion of whether Irwin is being responsible or irresponsible still having a class like this. It's a discussion of the biology. And I guess some epidemiology. Lindsay will do some epidemiology. But it's not a forum about Irwin. But it is about coronavirus, which you're all aware of. And coronavirus there's a likelihood, a possibility, probability, you pick, that coronavirus will impact Irwin soon. And so the question is, what are we going to do here? Well, if classes are canceled, or if the campus is shut down, obviously we won't have classes and we won't have lab. Classes are easy. I've been videoing classes from the start, and so I will continue that, and we will do online education as far as classes are concerned. It's not as good, but it's what we can do if classes are canceled. Lab is another story. If labs are canceled, we will also go online with labs. I will distribute worksheets, and I will distribute a way for you to do A&P revealed, but we won't be able to have face-to-face time with the cadaver. We'll figure it out as we go along. Exams are another story. So there is an exam scheduled for the week after break, and that will happen. How it will happen is a question. If we are still having classes, we will do it the way we did exam 1, where you'll have 3 options. You can either take it here in the morning, or you can take it in the lab in the afternoon, or you can take it in the lab in the evening of that Thursday, just like we did for exam 1, if campus is open and if we are having meetings like this. If we are not having meetings like this, or if campus is not open, it will be an online exam. I will make the exam in such a way that you won't write anything with a pencil or paper, a pencil or pen. You will do it on an electronic document, which means it'll all be text. It'll be short answer or short essay answers. And then you would create a Word or a PDF document and send it back to me electronically. There'd be no paper. You won't want to touch my paper, and I won't want to touch yours under those circumstances. So it'll all be electronic. So the point is, and for those of you watching from home, this is true, you want to take your computer and everything you might need to take an exam home with you over break. Because there's a chance you'll be taking this exam at home the week after break. So take your computer, make sure your computer can get to the internet, at least, Moodle, email. Those are YouTube for seeing these videos. Make sure, have your coloring book with you, have your textbook with you, have your notes with you. Take your materials home over break.
If that's the case, how much time would be allotted for?
So, so there's 2 kinds of exams. One is I just give you an exam that is closed book, and I put my faith in academic integrity that you will keep the exam, that you will do a closed book exam. Another way to do it would be to say, I don't need to do that. I'll do an open book exam, a problem solving exam. How I would hope, because it's at home, I guess I would feel that the time constraints are less severe, that you don't have some place to be at 4:30. But I would certainly not make an exam that was a 2-day exam. I would try to construct an exam that could be done in 2 or 3 hours. That would be my goal. Other questions about the near uncertain future. Email is our best friend here. Just, if you have a question, send me an email. I hope you know that I respond very quickly, and that's how we'll have a conversation. If it's something, if you ask a question that I feel, wow, everybody should know this, I will broadcast it on Moodle, so check Moodle with regularity. Okay, so that's Moodle, or that's what's coming up. Let's go ahead and get to work and finish the motor system. Because the plan is to finish the content of this section of the course today, have a break, recharge, regenerate, re-energize, whatever. Tuesday after break, if we are having meetings like this, would be a review and question and answer session. And and then hopefully we would have the regular exam on Thursday of the week after break. Oh, I meant to say, lab tonight. I will be having lab tonight, but some of you might be uncomfortable being in close quarters as we are in the lab. I don't know what's the most responsible, what's the best thing to do under these circumstances, really any better than you do. So I'm making lab tonight optional. If you are uncomfortable being in close quarters, then just don't show. You won't learn anything, but you won't get penalized. If on the other hand you think that that you're okay with doing what we do in lab, which is sitting at a computer and doing the worksheet and working on the cadaver, please come to lab, because I'll be there and I'll be happy to have lab with you. But the point is, it's optional. You make your best decision based on your health status, where you're planning to go, and the people that you'll be shaking hands with over break, etc. I trust you to be, to make your best decisions about that. Okay, motor systems, part 2. So what we're going to do today is talk about spinal cord, reflexes, brain control. So we're going to sort of work our way up the motor system from the skeletal muscle that you now are expert in, and get to the brain by the end of the class today. So we've been focusing on, previously we were here. Now we are going to go here, how motor neurons control skeletal muscle and the organization of motor neurons. Then we will talk about this circuit in the spinal cord that reflexes and patterns of movement. And then we will talk about the brain, how the brain impacts the spinal cord. This is our roadmap. I want to remind you that motor neurons are what cause muscle to contract, right? One motor neuron causes a set of muscle fibers to contract, and that's a motor unit. So the question that should have been in your mind from the start is, where are the motor neurons? And the answer is, the cell bodies of motor neurons are in the ventral horn of the spinal cord. So you hopefully remember that the dorsal horn of the spinal cord is where sensory neurons came in, and in the case of pain and temperature neurons, that's where they synapsed. And in the case of touch and pressure sensory neurons, they entered here, but then they immediately ascended in the dorsal column, ipsilaterally. So this is the dorsal horn where sensory neurons are going to come in, and this is these are the ventral horns where motor neuron cell bodies and dendritic, processes are. This is where the cell bodies and dendrites of motor neurons are, and then their axons go out the ventral route and go to the muscles that they are connected to. Which muscles each motor neuron is connected to was determined during development, during embryological and neonatal development. So we're not talking about development, so we're assuming that the neurons have gone to and synapsed with the correct set of muscle fibers. So this is a funny pink stain of a spinal cord, but this is the gray matter, the ventral horn gray matter, and this is the ventral surface of the spinal cord. This is ventral white matter, dorsal white matter. It's not white, gray, and white because it's been stained funny. You'll remember that the cell bodies of sensory neurons are in the dorsal root ganglion, but the cell bodies of motor neurons are in the ventral horn. So here's another image of the spinal cord with white matter and gray matter. Not really very informative, but here is here are cartoons, schematic drawings of the spinal cord at various sections. So here is a cervical spinal cord with the ventral horns and the dorsal horns at the cervical, thoracic, lumbar, sacral, and coccygeal levels. And predictably, the ventral horns of the cervical, lumbar, and sacral are relatively large compared to the ventral horns of the thoracic region, because these are the motor neurons that control the arms. And these are the motor neurons that control the legs. And these are the motor neurons that control the chest and the torso. But we have more muscles in our arms and legs. So we have more motor neurons that serve our arms and legs. And not only do we have lots more muscle in our arms and legs, we have a lot more motor units because we can do fine control. We can't do very fine control of our torso. We're pretty, it's strong, but it doesn't have fine control. So lots more motor neurons that control the arm and leg relative to the number of motor neurons that control the muscles of the torso and back. The ventral horn, where the cell bodies and dendrites of motor neurons are, is organized. There's a crude map of the body in the ventral horn. And it's a pretty simple map. Proximal muscles have their cell bodies medial. Distal muscles have their cell bodies lateral in the dorsal horn. Proximal muscles, like the biceps and the triceps, the cell bodies of the motor neurons that innervate those muscles are medial in the dorsal horn. In the ventral horn. Did I say dorsal before? In the ventral horn. Let me do it all over again. Start from scratch. The cell bodies of motor neurons that innervate proximal muscles, like in the case of the arm, the biceps and the triceps, those cell bodies are located medially in the ventral horn. The cell bodies of motor neurons that innervate the distal muscles of the hand and fingers, the hands and finger muscles are distal, these are distal and this is proximal. Those motor neurons are located lateral within the ventral horn. And so the same is true so this is illustrating arms and hand, but the same would be true of the legs and feet. Muscles that innervate motor neurons that innervate the muscles of the proximal leg, the quadriceps and the hamstring muscles, would be medial in the ventral horn of the lumbar and sacral regions. Motor neurons that innervate muscles of the toes, the extensor hallucis longus, big toe, you'll see it in lab if you come to lab. Big toe is distal. Those motor neurons, the motor neurons that innervate the distal muscles for the toes and the feet, are lateral in the ventral horn of the lumbar and sacral regions. Is that clear? So that's the distal, proximal distal mapped to medial lateral within the ventral horn. There's also a mapping of flexors and extensors. Not a very good map, but a slight map. Extensors are more ventral. The cell bodies of motor neurons that innervate extensors are more ventral in the ventral horn. And the cell bodies of motor neurons that innervate flexors are less ventral in the ventral horn. So you would say that so you could estimate where the cell body of the flexor digitorum is, and put it next to the cell body of the neuron that innervates the extensor digitorum. Right? They would be out here. The cell body of the neuron that innervates the flexor digitorum would be above, less ventral, and the cell body of the neuron that innervates the extensor digitorum would be more ventral. You could put a little asterisk where those cell bodies are likely to be relative to each other. So now we have talked about muscles, and we've talked about the organization of motor neurons in the ventral horn of the spinal cord. And now we're going to talk about pattern generators in the spinal cord. Central pattern generators. We're still in the spinal cord. And to do these central pattern generators, I want to talk about two reflexes that you've experienced, that you're familiar with. So one of them is the knee-jerk reflex. And I think I printed these out big on page 2 of your handout so that you could annotate. Did I do that right? Yeah. Okay. So let's think about the organization of the extensors and flexors of the knee, right? Extensors and flexors of the knee. Extension of the knee, flexion of the knee. Flexors of the knee, extensors of the knee. Here's the kneecap. So this is posterior, this is anterior. There's a reflex. If you stretch the patellar tendon, which physicians or nurses do by hitting it, stretching it very, it's not a big stretch, but it's a very acute stretch. It's a very sharp stretch. A stretch of the patellar tendon leads to a stretch of the extensor muscles, let's say specifically the the rectus femoris. Rectus femoris is one of the quadricep muscles that causes extension of the knee. So do the vastus muscles, the vastus lateralis, the vastus medialis, and the vastus intermedius. Stretch of this tendon causes stretch of this muscle, because they're all sort of connected here through tendons. Stretch of this muscle, stretch of this tendon, stretch of this muscle, There's stretch receptors in the muscle spindles of this muscle. And that is a sensory input, a proprioception, sensory input to the spinal cord. So here we see in green the cell body in the dorsal root ganglion of a sensory neuron that innervates a muscle spindle. Activation of the muscle spindle by stretch. Action potentials propagate in the nerve, enter the spinal cord in the dorsal horn. These are proprioception. It's not pain, it's not touch, it's stretch, it's proprioception. And so one of the things that it does is it goes to the brain in the dorsal column. And you know that this isn't complete, because another of the things that it does is it synapses with interneurons, and the information goes to the cerebellum via the spinocerebellar tracts. Okay, so that's that part of it. The part to the brain is not fully shown here. But that's not what we're focusing on. We're not focusing on the brain, we're focusing on the reflex that is in the spinal cord. So now let's look at this organization here. This neuron, in addition to sending information to the brain, either directly by the dorsal column or indirectly by the spinocerebellar tracts, makes synapses in the ventral horn. It makes excitatory synapses with motor neurons that innervate the extensor muscles. That's this pathway right here, green to A and C. These are motor neurons in the ventral horn that innervate the extensor muscles. So you stretch the patellar tendon. I can't do it when I'm standing like this, but you stretch the patellar tendon and it causes a jerk by contracting the extensor muscles. But in order for there to be a jerk, the extensor muscles have to contract and the flexor muscles have to relax. If I contract both my extensors and my flexors, I don't get a jerk. So that's where this C comes in. The input activates an inhibitory interneuron that inhibits the motor neurons in the ventral horn that innervate the flexor muscles. So there is excitation of the extensors and there's inhibition of the flexors by this single small sensory input. So now we can tap the kneecap, stretch the muscle, and get a full-fledged knee-jerk reflex because we're contracting the extensors and relaxing the flexors. This reciprocal control of flexors and extensors is an essential piece of the central pattern that we're going to be generating. When you move a joint, you contract one and relax the other. Reciprocal control. When I extend my elbow, I am contracting the extensor muscles and relaxing and letting stretch the flexor muscles. When I flex my elbow, I am contracting the flexor muscles and relaxing and letting stretch the extensor muscles. That's the important thing. And this is reciprocal control that is caused by a fairly simple circuit in the ventral horn of the spinal cord. We don't have a flexor reflex, but the same thing is true for a flexor. When the system says flex the flexor, it's got a built-in mechanism to say relax the extensor. Reciprocal control. Okay, so far we've just got a simple reflex that's really boring. We're not at a central pattern generator yet. Now let's make one a slightly more complicated reflex, the pain reflex. You probably are aware that if you put your hand on a hot stove, your hand is back before you even know it. You have a reflex, a withdrawal reflex. You touch a hot thing and you reflexively, even before your brain knows it, you reflexively withdraw from the pain. So this is the cartoon here. It's not a hand withdrawal, it's a foot withdrawal. So let's walk through this. Painful stimulus. Pain, action potentials up the, let's say this is part of the femoral nerve. Now we change scales. Here is the dorsal root ganglion of this that has the cell bodies. Over here, just to make these fair. Here's the dorsal root ganglion. That's where the cell bodies of these sensory neurons are. So here's the green sensory neuron going all the way to the spinal cord. If this is in the foot, it's entering the spinal cord in the lumbar or sacral region. It's forming synapses in the dorsal horn. Pain pathway. Some of that information is crossing over and going to the brain on the contralateral side. It's pain information. It's hard to see because we've got all these colors, but it's this fiber right here, the one that makes sort of the U-turn. Okay, that's going to the brain. That allows us to sense the pain, but that's not what we're talking about here. We're talking about the reflex that happens even before it gets to the brain. So let's walk through this. Let's look at the ipsilateral side first. The pain activates through the green pathway the motor neurons in the ventral horn that innervate the flexor. And it inhibits through the inhibitory interneuron the motor neurons that innervate the flexors. So you step on a tack and you contract the flexors and you relax the extensors to withdraw. Put your hand on a stove, you reflexively contract the flexors and relax the extensors. That's what we've just done. It's the opposite of the knee-jerk reflex, but it's another hardwired reflex. You don't have to think about it. In addition to this ipsilateral reflex, we have a contralateral reflex that is stimulated by the same stimulus. The same stimulus that did this on the ipsilateral side, contracting the ipsilateral flexor and relaxing the ipsilateral extensor. There are neurons that cross over. There are excitatory pathways that excite the contralateral extensor and inhibit, through this inhibitory interneuron, the contralateral flexor. So we contract the ipsilateral flexor, and we contract the contralateral extensor. Why do we do that? Well, you don't want to fall over. You step on a tack, we have evolved mechanisms where we can reflexively lift one foot and reflexively contract muscles on the other side to avoid falling over. Now we're starting to see a central pattern. Reciprocal control ipsilateral, flexors and extensors, flexors and extensors, flexors and extensors. Reciprocal control contralateral, flexors and extensors, and opposing reciprocal control, ipsilateral versus contralateral. So when I flex my right knee, I reflexively, when I flex my right knee, I reflexively extend my left knee. And this helps us not fall over, but it's an important part of what's going to become locomotion. When I flex my right knee, I extend my left knee. When I flex my left knee, I extend my right knee. When I flex my right knee, I extend my left knee. You see, I'm starting to build locomotion. That's where we're going. This simple pattern generator is a step towards being able to walk. Now, I can't do the whole motor control pathway for walking. It's too complicated. But this makes it at least plausible that you can imagine, okay, there's thousands of neurons here. We're only looking at one or two at a time. It doesn't take much to say, okay, when I make 1,000 of these neurons synapses, I can do this reciprocal control. Flex on one side and extend on the other side. And so you might ask, in the pain reflex, when I flex on one side, do I automatically extend on the other side? I don't know if we do or don't, but it would be plausible that we do. Question, Austin?
That was my question. Is there an example in which pain stimulus occurs in the arm or hand, and we extend the contralateral limb?
I don't know.
Okay.
I know that it happens in locomotion. We don't walk on our hands,
Right.
and so our ipsi contralateral, we have reciprocal control of an arm. We have reciprocal control of an arm. We don't have as well developed reciprocal control of our right and left arms because we don't walk on our arms. If we were quadrupeds, we sure would. So we're getting to locomotion. So one of the simplest animals to study locomotion are lampreys. They have a spinal cord. They have a well-organized spinal cord. They have motor neurons in their spinal cord. And you know how a lamprey, it's a fish. It swims by making a sinusoidal shape. Well, it has a well-organized pattern of contraction. When these muscles are contracted, causing the animal to bend in this direction, these muscles are relaxed, allowing the animal to bend in this direction. When these muscles are contracted, these muscles are relaxed. And the timing of pattern of going from contraction here, works its way down the muscle, so down the animal. So it contracts 1, and then 2, and then 3, and then 4, and then 1, and then 2, and then 3, and then 4. This is the contractile muscles in an organized temporal sequence that allows the animal to do this smooth swimming motion. Okay, well, we're not really interested in lamprey. We're interested in human beings, and we have the same sort of thing. Pattern excitation of flexors and extensors that give us the ability to walk, to locomote. So what is the point here of this slide? Let's start here. Let's say this is the lumbar spinal cord. This is just a bubble diagram of the lumbar ventral horn. So we have motor neurons in the lumbar ventral horn that are controlling the legs. And when flexors on the left side are contracting, flexors on the right side are relaxing. And when flexors on the right side are contracting, flexors on the left side are relaxing by the same sort of thing that we saw back here. Reciprocal control on the left and the right side of flexors and extensors. We have learned a pattern. When I want to walk at a certain pace, I have my brain is telling my spinal cord, walk slowly. And then my spinal cord is initiating the program, the central pattern, to walk slowly. Flexing on one side and extending on the other, then extending on one side and flexing on the other, etc., etc. Locomotion. So there's the motor neuron output, whether it's, it could be excited or it could be inhibited, right? Because we need to relax the muscles that are not contracting. Left-right coordination, alternating left and right. Rhythm generating, that is probably coming from above. And that's how we get a pattern generator for walking in the legs. Now, intersegmental coordination. What does that mean? Lumbar to cervical. Information going from the motor neurons that control the pattern of contraction of muscles in the leg, informing the muscles, the neurons that control the muscles in the arm. We're not quadrupeds, but we evolved from quadrupeds, and you're aware that when you walk, your arms swing. And it's not just swinging because your body is moving. There's small amounts of muscle contraction that are causing the muscles to swing. And so when I'm stepping forward, I'm trying to not think about it, which is not so easy, but when I'm stepping forward with my left leg, my right arm swings forward. The flexors of my right arm contract. And when I step forward with my left leg, my right arm swings forward. And so there's forward and backward movement of the arm, and it's not just passive, there's muscle movement, forward and backward movement of the arm. We don't move our elbows very much. I mean, I guess if you were really working at it, you would flex your muscles. I mean, flex your elbows too, right? I mean, when you're running, that's a form of locomotion for sure. And I'm not going to do it because I'll look silly, but I'm looking silly enough, right? But we have reciprocal control of our shoulders and our elbows, even though we're not quadrupeds. And that's due to this intersegmental coordination. So full locomotion is a coordination of flexors and extensors reciprocally. Left and right reciprocally. And intersegments reciprocally. If I force myself, I can probably, I try to do this every year and I always fail. I can probably force myself to walk by putting my right arm, I can do this. I can force myself to walk wrong, but I have to really think about it. So my brain can override these patterns, but if I don't think about it, I do it like this. How do I do it? I do it like this. Right arm swings forward when the left leg is swinging back. There's a YouTube video of a cat undergoing the cat's three patterns of locomotion: walking, trotting, and galloping. And this cat has no brain. So those patterns of walking, trotting, and galloping, it's on a treadmill. And the treadmill is determining how fast the animal has to locomote. But it's all spinal. There's no connection between higher brain functions and the spinal cord in this animal. I'm not going to show it because it can be upsetting to see a cat that is brain dead walking. But if you're interested in, and it's an ancient video, so the quality is terrible. But if you're interested in seeing a cat walk, trot, and gallop with no brain activity, only spinal cord, go ahead and connect to that YouTube video. Yeah.
So, like, how would that apply with humans and, like, people that regain, like, walking abilities after spinal cord injuries?
Yeah.
Is it, like, the brain does have...
So, when someone has a spinal cord injury, typically their spinal reflexes have been disconnected from the things that tell them to go. And so if we can reconnect the brain to the spinal reflexes, we can reconnect motor control. So it's not that the spinal circuits are dead, although sometimes they are. What's often dead is the on-off switch. The ability for the brain to say, okay, spinal circuit, go. Walk, run, walk fast, walk slow, run fast, run slow, stay still. All that information from the brain to the circuit, to the central pattern generator is what is cut off. And the central pattern generator doesn't do anything without being told to do something. So a lamprey that's had its spinal cord injury, all of these neurons here are still functional, but the lamprey is paralyzed from the injury down. Eventually, in lamprey, it turns out that, and this is my summer research, eventually it turns out that these neurons in the lamprey reconnect, and 3 months later, a paralyzed lamprey is swimming normally again. We don't have very much of that ability. Was there a question over here? Yeah, Dawn?
Yeah, I was just wondering about like individual variations, you know, walk a little bit differently. What level does that occur at?
I don't really know. Probably at the spinal circuit, you know, at the pattern generator, but it could be in the brain. And it could be developmental, it could be some learned process. I don't know whether, if someone who walks funny or someone who walks one way, did they learn it from their mother and father, brother and sister, or were they born with something intrinsic that developed into that way of locomoting? I don't know. Remember, a lot of this is learned, right? We're not born knowing how to walk. Not only do we not have the balance, but we don't have the circuit. We haven't developed the circuits yet. We have the circuits for reflexes. Babies have a pain withdrawal reflex. Babies have a knee-jerk reflex. They just haven't developed the pattern generators necessary to walk. And nor have they developed the balance and control necessary to walk. Okay, so where are we? We've got our muscles. We've talked about motor neuron organization. We've talked about central pattern control of motor neuron, central pattern organization of motor neuron excitation and relaxation that leads us to locomote, to walk or run. And now we're going to go into the brain. We're going up here. We're going to go to the brain. The brain that tells these central pattern generators go fast, go slow, stop, whatever. So let's do the cerebral cortex and then we'll take a break. So here is the brain, and superior view, anterior, posterior, left, right, longitudinal fissure, central sulcus, frontal lobe, parietal lobe, occipital lobe. Remember, this is the somatosensory cortex, which has a map of the body on it. Now we're in the motor cortex. The motor cortex is the most posterior part of the frontal lobe in the precentral gyrus on both hemispheres. So a sort of oblique sagittal section, no, coronal section of the brain here. Midline, right? This is along the central, longitudinal sulcus. Here's lateral. Here's temporal lobe. Here's the lateral sulcus. Just getting oriented, right? This is a nearly coronal section through the motor cortex. And there's a map of the body there. Head and face muscles, lateral. Tongue muscles, real lateral. head and lips and face muscles lateral. We have real fine control of our tongue, of our face. And so there's a lot of neurons that are dedicated to controlling those motor neurons. Remember, these are not motor neurons. These are neurons that control the motor neurons, which are the sort of output from the central pattern generator. So head and face is lateral. Hand, lots of neurons dedicated to controlling hand movements, is sort of superior lateral. Arm and trunk and legs, superior. And then as we go into the longitudinal fissure, medially, we get to the neurons that control, the motor neurons that control the toes and the feet. So, and luckily, this is pretty much the same map as we saw in the somatosensory cortex. We had guts out here, but we don't control the movement of our guts. But we do control the movement of our tongue. Head and face was pretty much like this in the somatosensory cortex. Hand and arm were pretty much like this in the somatosensory cortex. All of this looks just like the somatosensory map. The somatosensory map has genitals here. We don't have conscious control of our genitals, so it's not there. So the motor map and the sensory map are kind of aligned. The neurons of the motor cortex make axons because they're going to connect with the body. Those axons go through the internal capsule and then go through, and most of them cross in the medulla. So this is white matter coming from the motor cortex. And that's most of what was inside the internal capsule. Internal capsule is white matter from the motor cortex. Here's a colorful cartoon of those axons. This is not a cartoon. This is actual data. This is from a imaging modality called something tensor analysis. So this is actual data showing the axons of the motor cortex. Diffusion tensor imaging is what it's called. It actually images the water molecules inside the axons. And because the axons are so fine, the water molecules are the movement of the water molecules are coordinated. And it's picking up the coordination of movement of water molecules. It's pretty incredible. Let's stop here, we'll take a break. So I want to talk about the organization of the cortico-spinal connections, the connections between the cortex and the spine. That's where we'll go. Let's take a short break to read it. Just like there were some sensory paths that I wanted you to learn about, their localization and what information they carry, that are blue in this cartoon. Remember, we have the anterolateral column that's carrying contralateral pain and temperature information, the dorsal column that is carrying ipsilateral touch pressure and proprioception, and the spinocerebellar tracts that are carrying mixed and ipsilateral and contralateral proprioception information to the cerebellum. That's what I wanted you to know in blue. Yeah?
Could you explain like why dorsal column is ipsilateral even though it ends up on the other side of like it crosses over eventually?
It crosses over eventually in the brainstem.
Mm-hmm. Oh,
Because
so
all of the information has to get to the contralateral cortex. So it has to cross over somewhere.
So it's ipsilateral in the spinal cord.
Ipsilateral in the spinal cord,
Okay.
crosses over in the brainstem. Thank you for the clarification. It all goes to the contralateral somatosensory cortex. But in the spinal cord, it's ipsilateral, because it hasn't crossed over yet. So that's the information I want you to know in blue. Anterolateral, spinocerebellar, dorsal, and the modalities that they carry. Now we don't have to worry about modalities, it's all motor in red. And there's really three-ish things I want you to know about the organization of white matter of the motor pathway. One is the corticospinal tracts, which are lumped together and they're called the pyramidal tract, but I'm going to call them corticospinal. Corticospinal tracts on the right and on the left. The lateral corticospinal tract and the anterior corticospinal tract. Corticospinal, going from the cortex to the spine. Lateral and anterior. And what are the other red spots? The other red spots are other red spots. There are other motor pathways that I want you to know exist, but I don't want you to memorize. You know, their names or what they do. They're interesting. Vestibulospinal. So we get information from our vestibular system directly into our bodies without going to our cortex. Vestibulospinal. Olivospinal. The olives are a nucleus in the medulla. So we have some other motor pathways that I want you to know about: the lateral corticospinal, the anterior corticospinal, and other. Now, how are they organized? 90% of the axons from the motor cortex are in the lateral corticospinal tract and cross over in the medulla. So the motor cortex of the right hemisphere mostly controls the muscles of the left body, the left muscles, because of the crossover. 90% of the lateral corticospinal tract, is crossed over. No, 90% of the axons from the motor cortex are in the lateral corticospinal tract and crossed over. 8% of the axons from the motor cortex are in the anterior corticospinal tract and are not crossed over. So the lateral corticospinal tract in the spinal cord is carrying information to that side of the body, but it's from the contralateral hemisphere of the brain. 8% of the information is in the anterior corticospinal tract, and information on the right side of the body is from the right hemisphere of the brain. So this motor information is from the left hemisphere, and this motor information is from the right hemisphere, because this was crossed over in the medulla, and this was not. Crossover in the medulla, no crossover in the medulla. The cortex is not shown here. The cortex is up on the second floor. Dolly?
The 8% in the anterior corticospinal tract, do they cross over at a certain point, or
No.
do they just go to
They go to the
ipsilateral?
muscles of the ipsilateral side. And so now if you're doing arithmetic, we have an unaccounted for 2%, and 2% of the axons from the motor cortex are in the lateral corticospinal tract, but they are uncrossed. So really, 92% of the information is in the lateral corticospinal tract. 2% of it doesn't cross, and 90% of it does. And you can probably predict what I think is important. 90% is important, 8% is important, 2% is less important. So focus on the important things. Yep.
Does the anterior corticospinal tract have any relation to CPGs? To
These are all of these are making synapses on CPGs.
Okay.
Some of them are making synapses directly onto motor neurons, but most of them are making synapses into a pattern generator synaptic network that is controlling motor neurons. So what we just talked about were axons from the cerebral cortex. I'm going to try to put this together. We were talking about axons from the motor cortex in the post-central gyrus, generating white axons, white matter, that went through the internal capsule into the medulla, and then some of it crossed over and some of it didn't. Most of it was in the lateral corticospinal tract, but some of it was in the anterior corticospinal tract. Okay, that's where we were. These neurons controlling these axons going through the internal capsule and making the lateral corticospinal tract, the anterior corticospinal tract, and other motor pathways. Now, they need to finish this by introducing subcortical regions that are essential for controlling movement. So what happens is that we have nuclei deep in the brain that tell the motor cortex to tell the central pattern generators to do their thing. Movement actually, the initiation of movement is deep. Initiation of movement, planning of the movement, central pattern generators, and the motor neurons doing the movement. Well, the muscles do the movement. The central pattern generators and the motor neurons, integrate and execute the movement. So you might remember that here's a cartoon, thalamus, lateral ventricle, septum, corpus callosum. In this slide, it's called the basal nuclei, caudate, putamen, globus pallidus. We're deep in the brain. In this cartoonish section, we're seeing both the thalamus and the basal nuclei. Let's play this game. Who wants to start? Point to something and give me a name. Okay? Corpus callosum. Okay. Oh, okay, good. So,
Third.
yeah, I'll buy that. Third ventricle. Okay? It's not labeled, it's not,
Oh.
it's the,
How
you're right.
about like cingulate gyrus?
Okay, good. Cingulate gyrus. I'll give you some, I can give you a suggestion. So what is this white matter here? Okay, if you don't know that one, I'll give you another suggestion. What is this gray matter here? Or this gray matter here? Or this gray matter here, actually? Any of those?
Would that be the caudate nucleus?
Yeah, that's the caudate nucleus. Who's got those lists? Oh, okay, awesome.
Thalamus?
Yeah, sure, let's go with thalamus here. I'm not 100% sure I'm getting it right, but that's a good guess. And let's go with thalamus here too. Right? This is the thalamus. Yeah, internal capsule. So this white matter, remember, so this is the white matter coming from the motor cortex, going through the internal capsule. Those axons make the internal capsule. Let's see. Let's find, so these, what's this, is that the one you wanted?
Yeah.
Okay, butane, yes, that's correct. Okay, let me suggest one. What are these things here? Okay, let's try another one. What is this here? Medial temporal lobe, memory. Hippocampus. You got the first. Don't confuse the hypothalamus and the hippocampus. We cannot see the hypothalamus here. This is the hippocampus.
The lateral ventricle.
Yes. So that's the lateral ventricles. Those are called the lateral ventricles.
The fornix?
Okay, yeah, so now let's see, let's go, let's do this one. This is the last one I really want to get labeled. Do you know what these two structures here are? That's labeled by this one name. The name is actually given in the previous slide.
Globus pallidus?
Yes, that's the globus pallidus. So let's see if we get this right. One at a time to see if we got it right. Globus pallidus, hippocampus, third ventricle. Okay, we're batting 1,000. Thalamus, putamen, internal capsule. Okay, we didn't label this one, but that was also a piece of the thalamus. So all of this stuff in here is thalamus. Lateral ventricle, corpus callosum, cingulate gyrus. Yeah, we did it, we're good. So as a group, we get 100%. But remember, you don't get to work as a group. So this answer key is on movement. What's in yellow are the parts of the brain that I want you to know for the motor system. The hippocampus is not part of the motor system. The corpus callosum is not really part of the motor system. What's in yellow is the motor, are the subcortical structures that are important for the motor system. So here's a cartoon that simplifies what we're talking about. Cortex, white matter from the cortex, internal capsule labeled, caudate, putamen and globus pallidus, and deep in the midbrain is the substantia nigra. So substantia nigra, remember, you've heard this label before. In what context did we hear about substantia nigra? Dopamine. Now we're putting the dopaminergic neurons of the substantia nigra into a functional role in the motor cortex. We're also going to be talking about some thalamic nuclei. So remember, in sensory input, a lot of sensory input was processed in the thalamus before it went to the sensory cortex, whether it's the somatosensory cortex or the visual cortex or the primary auditory cortex or the taste cortex. A lot of that information was processed in the thalamus. Similarly, the thalamus processes motor information and integrates it and helps with the initiation and planning of movement. So I could ask you, what part of the thalamus is most important for processing visual information? There's a name of the nucleus in the thalamus that is most important for processing visual information. You might remember, even if you haven't studied it. The lateral geniculate nucleus. And there's another one called the medial geniculate nucleus that is important for auditory processing. And in motor, they're called the ventrolateral nuclei of the thalamus. You don't have to know that. I'm not going to ask you that. Okay, so what do we have here? This I don't want you to memorize, so you can actually put your pens down because I'm not going to ask you to reproduce this. But this is what explains Parkinson's disease. So initiation of movement starts in the substantia nigra. It tells the basal nuclei to do stuff, which including the ventrolateral nucleus of the thalamus, which tells the cerebral cortex to plan a movement, which then generates output that tells the central pattern generators do it. It being walk or run or crouch or jump or whatever it is the central pattern generator is adapted for. Okay? So it starts in the substantia nigra. So your brain is actually working a few scores of milliseconds, 20 to 100 milliseconds, before your conscious brain is telling your body to do something. You have unconscious planning and initiation steps before you're conscious of making the movement. So there's this funny delay where you're not even aware, but you're already planning the movement. Starts in the substantia nigra. These, blue is excitatory and red is inhibitory. There's a series of, there's excitatory and inhibitory dopaminergic, these are all dopaminergic paths from the substantia nigra, dopaminergic input to the putamen, which then sends inhibitory input to the globus pallidus, that generates input to the thalamus, that generates excitatory output to the cortex. So the start is substantia nigra, the output is excitation to the cerebral cortex, and then, and then there's output from the cerebral cortex that activates the central pattern generators. So watch what happens. There's a concept here that I do want you to understand, and that is double negatives. Activation of an inhibitory neuron that inhibits a second inhibitory neuron. Inhibition of inhibition, disinhibition, allows excitation. Inhibition of inhibition allows excitation. So excitation of inhibition is excitation. Inhibition of, let's count the number of inhibitory interneurons in this pathway. 1, 2, 3. There's an excitatory step, but there's 3 inhibitory path steps in this. So it's an odd number. So this is, but okay, 1, 2, 3, 4 is an even number. This inhibitory pathway also leads to excitation from the thalamus to the cortex. But it's inhibition of inhibition of inhibition of inhibition leading to excitation. In Parkinson's disease, these neurons die. So we can go through the details here, but the lack of input leads to the lack of output, and initiation of movement is compromised because of the loss of dopaminergic neurons from the substantia nigra. And it compromises both the simple, so let's walk through this. Lack of excitation, lack of inhibition, so uncontrolled inhibition leads to uncontrolled inhibition of the output. So loss, and we can do the same thing with the other pathway. So what do I want you to know? I want you to know this concept that inhibition of inhibition, or disinhibition, is in fact an excitatory thing. And dopaminergic neurons from the substantia nigra to the basal nuclei control initiation of movement. And I'm happy to walk through this in more detail, but that's enough for me. Now, I would be delinquent if I didn't mention the cerebellum here, but I'm only going to mention it. Because the cerebellum is home, well, A, it's because it's home of the most beautiful neurons in the world, the cerebellar Purkinje neurons. Look at this dendritic arborization of the cerebellar Purkinje neurons. So it's just amazing. They are the most beautiful neurons ever. And so here's the dendritic organization of a cerebellar Purkinje neuron, and here's the output axon. This is the thing that's filling this neuron with a fluorescent dye. So you have millions of these in your cerebellum, and maybe billions of them in your cerebellum, and they each have about 10,000 synapses. So your cerebellum is doing a lot of synaptic processing to coordinate movement. And and again, you don't have to know hardly any of this, but this is an organization of the cerebellum. So here's the cerebellum, here's the motor cortex. The motor cortex informs the contralateral cerebellum, which was getting information from the spine. So the cerebellum is integrating, the left cerebellum is integrating information from the left side of the body from the spinocerebellar tracts, proprioception, and integrating information from the motor cortex that is mostly controlling the contralateral side of the body. The cerebellum informs the contralateral cerebellar cortex. So there's input and output coordination. Input from the cerebral cortex, input from the body, integration, planning, coordination, output to the cerebral cortex that then does output to the body that we've talked in the corticospinal tract that we've talked about. That's really all this slide is trying to get you to know. It's got some great names that I don't care. Flocculonodular lobe. Just love saying that word, but you don't have to memorize that. What do I want you to know? That the cerebellum is part of coordinating motor control. If you don't have a functioning cerebellum, you can still walk because you've got a central pattern generator and you still got a cerebral cortex that turns on the cerebellar cortex, or turns on the central pattern generator. But your walking is going to be stilted and unbalanced, and it's not going to be smooth and secure like it would be if you had a functioning cerebellum. Okay, that's it for today. That's it for this week. Have a great break. Keep your eyes on email and Moodle for news about what happens after break. You're invited to come to lab, but if you don't come to lab, it won't cost you anything.